Hello, everybody. Welcome to Ms. McGuire online video lecture. Today we're covering human genetic cells. So let's go ahead and begin. Learning outcomes for this chapter. After completing this chapter, you should be able to explain why it's important to know the cellular basis of a disease, distinguish somatic cells from germ cells and diploid and haploid cells, explain how cells differentiate, list the 4 major chemicals in cells, describe how organelles interact to secrete, use energy, and clean up debris, describe the structure and function of biological membrane, list the component of the cytoskeleton, list the characteristic of a stem cells and a progenitor cell, and describe the 3 main sources of human stem cells. Introducing to the cells. Inherited traits, quirks, and illnesses arise from the activities of cells. And actually, how well your entire organism functions depends on function of your individual cells. Cells are the basic units of life and comprise the human body. Our body includes more than 290 specialized or differentiated cell types. We have billion of cells, right? But out of this billion cells, we have about 290 varieties of cells. These aggregate and interact to form the 4 basic tissue types. So cells form tissues. And in human body, you have 4 tissue types: epithelial, connective, muscle, and nervous tissue. All cells share certain features, but they also specialize because they express different subsets of genes. Now, we all start with as a single cell. So when father sperm fertilizes mother egg, the first cell that is formed is zygote. Now, human zygote has like 46 chromosomes. So all your cells, your entire body is made from this very first cell. That means that every cell in your body has the same genes, same set of DNA, but still cells are different. Of course, your skin cells are not the same as your neurons, not the same as your liver cells. Why? If they have the same DNA, how they can look and act and function differently? But that's because they express different genes. So in your skin cells, only those genes are expressed that are needed for function of skin cells. And in your neurons, only those genes are expressed that are needed for function of neuronal cells. So here's connective tissue is a variety of cell types and surrounding materials that protect, supports, bind to cells, and fill spaces throughout the body. Connective tissue includes cartilage, bone, blood, and fat. Epithelium or epithelial tissue, tight cell layers that form lining that protect, secrete, absorb, and excrete. Muscle cells, cells contract, providing movement. Nervous cells, neurons transmit information and electrochemical impulses that coordinate movement and also sense and respond to environmental stimuli. And nervous tissue also made from supporting cells called neuroglia that support and nourish neurons. So that's a 4 major tissue type of human body: connective, epithelial, muscle, and nervous. Somatic cells are also called body cells. They have 2 copies of the genome and are said to be diploid. Germ cells are sperm and egg cells. Another name for egg cells is ova. Ovum is one, ova are plural, and they have copy of genome and they are haploid. The meeting of sperm and egg restores the diploid state. So if you made of cells, right? So here's your cells. All the cells, they are 2n. So they are diploid. And for human cells, 2n equals 46. So we have 46 chromosomes. Now, if you have a sperm, it has only 23 chromosomes. An egg has 23 chromosomes only, so they are haploid. So when they unite, we restore the ploidy, because when they unite, we end up with 46 chromosomes again. So sperm and egg has, just one N, so they haploid. And when sperm fertilize egg, we go back to 2N, 2 diploid cells. Stem cells are diploid cells that divide to give rise to differentiated cells and to other stem cells in a process called self-renewal. Now, to be a stem cells, they must have two properties. First, they need to be able to differentiate into different cells. And second, they need to self-renew itself. All right, so let's look more into that. So biologists, we will get to this. So at the end of this PowerPoint, we will go back to stem cells and we will, we will see why and how they can self-renew and how they can differentiate. Okay. Now, biologists recognize three basic types of cells that define three major domains of life. Now, our entire life on this planet is divided into three big groups, and they call domains, right? So one domain is archaea, another is bacteria, the unicellular prokaryotes. Unicellular means they're made of one cell, and prokaryote means they don't have nucleus. So they have cell and they have DNA. So DNA is inside cytoplasm, so no specific organelle, so they prokaryotes. And domain number 3 is eukarya. Include both unicellular and multicellular eukaryotes. And eukaryote means those cells have nucleus, and DNA mostly is found inside the nucleus. Cells of domains contain a globular assemblies of RNA and proteins called ribosomes. Now, all these cells, archaea, bacteria, and eukarya, they have ribosomes. Ribosomes are made of RNA and proteins. Ribosomes are essential for protein synthesis. Eukaryotes may have arisen from ancient fusion of bacterium with archaea. So that's a beautiful picture over here. Now look, what do you see here? This cell is white blood cell. So that's a cell of your immunity called macrophage, right? So that's your cell. So it's a eukaryotic cell. And look what it does. It's eating up bacteria, right? So that's what scientists think. What if, it was actually symbiotic relationship between bacteria and archaea, or some maybe similar cells, not necessarily those, but what if somehow they get into a point when the bigger cell engulfed a smaller cell, right? And then how we get eukaryote. But again, that's a theory. We not sure if this is how it happened. Just a second. Yeah, okay. So let's continue. Cell chemicals constitution. The major macromolecules that make up cells are carbohydrates, sugars, and starches. Well, just make sure that starch, that's in plants, Okay, in the animals, we have sugar and glycogen. We do not have starch. But carbohydrates include starches, and you find starch in plants. Lipids, fats and oils, proteins, and nucleic acids, DNA and RNA. Now, carbohydrates provide energy and contribute to cell structure, right? Of course, when we talk about cell structure made of carbohydrates, we're mostly referring to plants and fungi. Lipids. Lipids form bases for some hormones. We have lipid-based hormones. We call them steroid hormones. Lipids form membranes, provide insulation, and store energy. Proteins have many diverse functions, such as forming contractile fibers in muscle cells, enabling blood to clot, forming the bulk of connective tissue. Enzymes are proteins. They are especially important proteins because they facilitate or catalyze biochemical reactions. Nucleic acid, DNA, and RNA are the most important macromolecules to study of genetics. Structure of a cell. Cell surrounded by plasma membrane, also called cell membrane, and contains intracellular organelles. Cells contain cytoplasm, stored proteins, carbohydrates, lipids, pigment molecules, and other small chemicals. Now, the cytoplasm is made of cytosol and all the organelles. So pretty much water that called cytosol and organelles make cytoplasm, and then you have all other molecules listed over here. So here's a diagram of simplified eukaryotic cell that's surrounded by plasma membrane, has a nucleus, and all these organelles inside a cell. Organelles divide labor by partitioning certain area or serving specific functions. They keep related biochemicals and structures close to one another to interact efficiently. Function of organelles include enable a cell to retain and use its genetic instructions, acquire energy, secrete substances, and dismantle debris. Okay, we're going to start with nucleus. The most prominent organelle surrounded by a layer called nuclear envelope and contains nuclear pores, that's just a channel that allow movement of biomolecules, nuclear lamina that provides mechanical support and holds nuclear pores in place, nucleolus where ribosomes are produces ribosomes, they assemble inside nucleolus, and other components such as chromosomes, very important, RNA, and the fluid portion is called nucleoplasm. So here we can see a cell over here, right? And the most prominent organelle is nucleus. So the nucleus itself has a nuclear envelope. It's a double membrane with nuclear pores that allow molecules to move in and out. Inside the nucleus, you will have nucleoplasm and chromosomes, or chromatin actually. And there is another part over here called nucleolus that we're going to talk about. Okay, what is this? Oh, we already, let me just mention, this is this dense part inside the nucleus is nucleolus, and that's where ribosomes are assembled. So, so here's the example of or illustration of how organelles work together to produce something. In this example, to make milk. Secretion illustrates how organelles work together to coordinate the basic life functions. So we're gonna have a nucleus. Inside the nucleus, you have DNA that encodes milk proteins and certain enzymes and transcribed it into messenger RNA. So the DNA is inside the nucleus. Then we make a copy of specific gene, and it's called mRNA. So here's mRNA leaves the nucleus. mRNA exits through the nuclear pore, right there. Then mRNA moves to surface of a rough ER, so that's organelle is rough ER, those are ribosomes, where proteins are synthesized on ribosomes using amino acid in the cytoplasm. Then lipids are synthesized in a smooth ER, so this organelle is called smooth ER. Sugar are synthesized and proteins, sugar also synthesized inside smooth ER. Then proteins and sugar move to Golgi apparatus. From the Golgi apparatus, the secretory vesicles are formed. They move toward the plasma membrane and secreted. And also, you know, milk has proteins and sugar and fat. Now, fat is synthesized separately in a smooth ER and moved through the vesicles in the lipid droplets, so they also exit the cell. Right, so here what we have. So everything that I explained you listed on this slide. And right, so here. So just pause the video and read it again if you need to. Endoplasmic reticulum, or ER, is interconnected membranous tubules and sacs. ER winds from the nuclear envelope to the plasma membrane. We have two types of ER. Rough ER contains ribosomes and involved in protein synthesis, because if it has ribosomes and ribosomes synthesize proteins, then rough ER also involved in protein synthesis. Smooth ER does not have ribosomes. So if it doesn't have ribosomes, it cannot make proteins. Then it's important in lipid synthesis. Proteins exit ER in membrane-bounded sac-like organelles called vesicles. So if you look over here, right there, yeah, it doesn't really show on this, but it will be vesicles. So here they only show vesicle from Golgi apparatus, but ER also form vesicles, and those vesicles moves towards the Golgi. And then Golgi makes its own vesicles called secretory vesicles. And ER, when ER makes vesicle, we call them transport vesicles. Golgi apparatus, sac of interconnected flat membrane enclosed sacs, processing center that adds sugar to lipids and to proteins, forming glycoproteins and glycolipids. Products are released into vesicles that bud off to the plasma membrane. Some cell types have just a few Golgi apparatuses, but those that secrete may have hundreds. Lysosomes are membrane-bound sacs containing 43 types of digestive enzymes. Dismantle bacterial remnants, worn-out organelles, and excess cholesterol. So they pretty much destroy all these parts. Engage in autophagy. Autophagy means eating yourself, right? So lysosomes destroy, if they destroy your own organelles that you don't need anymore, then that's what's called autophagy. Endosomes are vesicles that derive from the plasma membrane that ferry materials to lysosomes. For example, low-density lipoprotein, LDL cholesterol. So that moved inside these endosomes. So here we have endosome. So you see that's a plasma membrane. So this is inside the cell, this is outside the cell. So if you have endosome, so vesicle is formed with some substances, some extracellular debris, right? They moved inside the cell and they fuse with lysosomes. So here the lysosome is synthesized, right? Lysosome fused with the vesicle, and we pretty much will digest this extracellular debris, or if it's its own organelle, for example, mitochondria, we can break down mitochondria as well. So lysosomes are like garbage disposal of a cell. Cells differ in the number of lysosomes they contain. Macrophages, for example, they require many lysosomes to digest bacteria. Liver cells require many to break cholesterol, toxins, and drugs. Absence or malfunction of an enzyme causes a lysosomal storage disease. So remember we said we have, okay, let me go back. So we have like 43 types of digestive enzyme inside lysosome. Now, if some of these enzymes are not functioning enzyme, we call it lysosomal storage disease. For example, Tay-Sachs, that's inherited disorder. Nervous system become buried in lipids, causing death within 3 years of age. So you build up so much lipids inside your brain, and you don't have lysosomes to break down these lipids, and the brain stop functioning because it just covered in fat. Peroxisomes are sacs with outer membrane studded with several types of enzymes. So peroxisomes are also sacs with enzyme inside, but those are not digestive enzymes. Those actually oxidases, because the major function of peroxisome is detoxify components from exposure to oxygen-free radicals. They also, break down lipids, rare biochemicals, they synthesize bile acids. So they similar to lysosome in some way because they do destroy some molecules, but they use different enzymes. Peroxisomes are abundant in liver and kidney cells because liver is the organ of detoxification. Adrenoleukodystrophy is an example of genetic disease caused by absence of peroxisomal enzymes. So it's also affect brain. When we cannot break down chemicals and toxins, then they can damage our organs. Another organelle, mitochondria, provide energy by breaking chemical bonds that hold together nutrient molecules in food. So what mitochondria does, it takes food, for example, sugar, and it breaks the sugar and release energy to make these molecules called ATP, adenosine triphosphate. So the energy is stored in the form of ATP. Mitochondria is surrounded by two membranes. Now look, mitochondria is plural, mitochondria is singular. Now each mitochondria has two membranes. Inner membranes form folds called cristae that hold enzymes and catalyze biochemical reactions. Very important organelle. So here you can see the electron micrograph. This is all not real color here, but that's a mitochondrion. And you see those folding sites called cristae. So that's the inner membrane of mitochondria. Okay, so here's our summary of the organelle and their function. Endoplasmic reticulum, membrane network, rough ER, has ribosomes, smooth ER does not. Rough ER, site of protein synthesis and folding, smooth ER, lipid synthesis. Golgi apparatus, stacks of membrane enclosed sacs, site where sugar are made and linked into starches or joined to lipids or proteins. Proteins finish folding and secretion is stored inside Golgi. Lysosomes, cell containing digestive enzyme, degrade debris, recycle cell components. Mitochondria, two membrane, inner membrane enzyme studied, releases energy from nutrients from your food. Nucleus, porous double membrane sac, contains DNA, separates DNA within cell. Peroxisome, cell containing enzyme, break down and detoxify various molecules. Ribosomes, two associated globular subunit of RNA and protein. scaffold and catalyst for protein synthesis. In vesicle, membrane-bound sac, temporary storage or transport substances. Biological membrane. A biological membrane has a distinctive structure. It's composed of double layer, it's two layers, or bilayer of molecules called phospholipids. Phospholipids have a phosphate part, it's hydrophilic, water-loving, and two fatty acids that are hydrophobic part of molecule, water-hating. Proteins are embedded in a phospholipid bilayer. Also, membranes contain carbohydrates on their external surface. So we're going to look at the diagram in a second. Now, proteins that reside within the lipids, aboard lipids, contribute to cell identity, they transport molecules, they keep out toxins and pathogens. Some membrane proteins form channels for ions, channels so ions can move in and out. Channeler pathies, like it's a pathies means pathology. Channelopathies are diseases that that stem from faulty ion channels. So if those channels and channels are proteins do not function, we might have a disease. Example, cystic fibrosis that I explained in the first lecture. Okay, so here's the structure of cell membrane. So you see that's one layer. So here's one layer. And here's the second layer. So it's a double layer. And those molecules that has this round structure and then two tails, that's a phospholipid. So that's why we call it phospholipid bilayer. Inside, we will find proteins. And proteins can have different functions. They can be enzymes, they can be recognition molecules, or they can be channels, like shown over here. See how they have opening and sides, so molecules can, or ions can move in and out. Right, so that's a protein. Another part of membrane is this molecule, that's lipid, cholesterol. And those shown here in green, those are sugar. When sugar are attached to protein, we call it glucoprotein. When it's attached to lipid, we call it glycolipid. So it's either glycoprotein or glycolipid. Now here's phospholipid. It has a head. It's a phosphate group and it's hydrophilic, so it loves water, and two tails, hydrophobic, made of fatty acid. And because head loves water, they will be facing water. So we have water outside the cell and we have water inside the cell, and that's the orientation of this phospholipid allow us to form bilayer. Many molecules that extend from the plasma membrane are receptors, but they bind ligands that may set into motion a cascade of chemical reaction inside the cell. What it says over here, that here we have a cell and we might have a protein here, and the protein is a receptor. So another molecule can bind to this protein and cause some chemical reaction inside a cell. This is called signal transduction. Molecules form pathways that detect signals from outside the cell and transmit them inwards. So, for example, this ligand molecule can be growth hormone. Right? So the growth hormone attaches to protein receptor and sends signal to the cell to divide, so the tissues or organs can grow bigger, right? Cellular adhesion. Some plasma membrane helps cells to attach to other cells. So here we might have like one cell and another cell, and we will have protein between them that keeps them together, attaches them to one another. Cytoskeleton, a meshwork of protein rods and tubules that serves as the cell's architecture. positioning organelles, and providing overall 3D shape. Now, there is 3 types of elements that make cytoskeleton. Hollow microtubules made of tubulin, solid microfilaments, which consist of special protein called actin, and intermediate filaments. They are made of more than one protein type. Right, so here's part of the cytoskeleton, microtubules, right, like a bigger hollow filaments made of tubulin. Then this is our intermediate filament that made of several different proteins, and here's our microfilament made of actin molecule. And if you look inside the cell, so here's the cell, right? And it's not just kind of boring fluid inside. You see all these fibers, all these proteins of cytoskeleton that give cell its shape, allow substances to move inside the cell. So very, very complex structure. Some cells, they have what we call appendages. Like this one has appendages called cilia, and cilia can move. So this cilia is made of microtubules. And cilia, when cilia moves, it beats, it can move something along the surface of the cell. In this example, it's mucus. So in your respiratory tract, you have ciliated epithelial cells. Cilia are made of microtubules. They move mucus from your nasal cavity towards your throat, and then you swallow the mucus. And what mucus does, it cleans your airways. It traps bacteria or some dust, some debris in the air. And instead of all this bacteria and dust go to your lungs, you actually, cilia beads, send it towards your esophagus. So you swallow the mucus and pretty much digest and destroy everything that you trapped in mucus. Okay, oops. And now we will talk briefly about stem cells, because I will have a separate lecture about stem cells. Stem cells divide by mitosis and produces 2 daughter cells, or as stem cells and progenitor cells. So, well, here no difference, right? Some cells, many cells can divide. So if cells divide, it makes 2 daughter cells. But stem cell is different, because stem cells, it's gonna self-renew. It's gonna produce stem cell. So you never completely are out of stem cells, right? Because every time stem cells divide, you have a new stem cell. And the second cell is called progenitor cells. It might be partially specialized. Now, progenitor cells cannot be a stem cell anymore, right? It's cells that will differentiate into some specialized cells, right? But it cannot become a stem cell, at least if it's not induced. So progenitor cells do not have the capacity of self-renewal. So when progenitor cells divide, they make a specialized cells, so you don't have another progenitor cells. This is important to remember about stem cells. Not only they will differentiate, of course they will differentiate into specialized cells, but they also keep their population. They self-renew their population. And again, as I said, I will have a separate lecture on stem cells. Self-differentiate down a cell lineage of stem, progenitor, and increasingly differentiated cells. So from stem cell, we go to progenitor cells, and then we go to differentiated cells. Stem cells and progenitor cells are described in terms of their developmental potential. We have totipotent cells that can give rise to every cell type. Pluripotent have fewer possible fates, so they can differentiate into many cell types, but not all of them. And multipotent have only a few developmental choices. Many, if not all, of the organs in the adult human body have stem or progenitor cells. That's amazing, right? Because when we think about stem cells, we're thinking, oh, well, those are embryonic cells, right? But no, actually, almost all organs in the adult human body have stem or progenitor cells. They might be small amount of them, and that's why they're not that useful as embryonic cells. But our organs are capable of producing new cells and regeneration. Okay, so here we can see the diagram of the pathway to cell specialization. So here we have our egg and sperm, and here's our fertilized egg or zygote that is totipotent. Right? So, I mean, well, yeah. So it will divide several times, forming a ball of cells. They are totipotent cells. Right? So stem cells, you see how they can renew themselves, and when they divide and form progenitor cells. And then progenitor cells, they undifferentiated cells that can be differentiated into several cell types. You see over here, it can become, for example, a bone cell or connective tissue cells, or it can be different blood cells and platelets and skin cells. Right, so that's the pathway from the stem cells to individual tissue cells. There are three general sources of human stem cells. First, of course, embryonic stem cells, created in a lab dish using the inner cell's mass of an embryo. So to get these embryonic stem cells, we need an embryo. Then we have induced pluripotent stem cells or iPS cells. Those are somatic cells that was reprogrammed to differentiate into any of special cell type. So those are actually the just a regular cell of our body that were stimulated, and they kind of reversed back to the stem cells. In adult stem cells, they tissue specific or somatic stem cells. Now, those are found almost in every organ of your body, and they specific to the tissue where they reside, right? So three types: embryonic stem cells, induced stem cells, and adult stem cells. Researchers study stem cells to learn about biology and to develop treatment for a variety of diseases and injuries. So embryonic stem cells, so here's the summary. Inner cell mass of every two of very early embryo somatic cells, nuclear transfer into egg cell. So embryonic cells. Induced pluripotent stem cells. Genes or other chemical reprogram somatic cells nucleus. No embryo required. So that's just kind of like a regular cells that were reprogrammed to become stem cells. And adult stem cells, somatic cells that normally function as stem cells from any stage of development, from fertilized ovum through elderly. All right, so here how a stem cell can be used. So for example, we have a person who need a stem cell for a treatment. We can use a donor stem cells or iPS, your own stem cells that were reprogrammed, right? Then we can isolate. So if it's your own stem cell, we can isolate them and reprogram to a less differentiated state. So kind of turn the clock back when those cells were not differentiated yet. Then we grow them on a petri dish, and then we stimulate those cells to differentiate into blood cells, muscle cells, or nerve cells. right? And that can be used for treatment of the patient. So we infuse or implant, right, those cells. Or we can use the donor cells, right? Or we can use own cells unaltered. For example, let's say if she has like a skin burn over here, right, you can use another part of the skin that does have stem cells, and you can just transfer it to another area, and those cells will divide and replace cells that are damaged. Stem cells are being used in 4 basic ways: discovery and development of drugs, observing the earliest sign of disease, create tissue and organ for use in implants and transplants, or to study, so we can create organs, and stimulating stem cells in the body via the introduction of reprogramming proteins. Right, so that's our 4 application for stem cells. And that's it for this chapter. Okay, thank you very much for watching. I hope it was helpful. If you have any questions, you can leave your questions in the just underneath this video, under this video in the discussion section. Just somehow let me know, and I will try to help you as much as I can. So again, thank you for watching, and I will talk to you next time.